call these cohesive ends or sticky ends, right, for reasons that we'll see in just a minute here. So here we're generating a 5' overhang, here we're generating a 5' overhang. HHA1, for example, generates a 3' overhang, right? Here we have a little bit of extra 3' sequence that we'll end up with, and the same down here, a little bit of extra 3' sequence. This is also cohesive ends. So we have restriction enzymes that can generate either a 5' overhang or a 3' overhang. These are extremely useful for reasons that we'll see in just a minute. We have other restriction enzymes, like HAE3, which generate what we call blunt ends. Notice this one does cut straight up and down. Okay, so this does not have any extra 5' or 3' sequence, and these are slightly less useful. They're useful in a very specific context, right, that we're not really going to get into here. Okay, but these are the 3 ways that restriction enzymes can cut DNA, either with a 5', a 3' overhang, or a blunt end. Okay? Now, why do we use restriction enzymes? Well, notice that each of these restriction enzymes have unique restriction sites. They recognize different sequences of DNA. So we can look at the sequence of DNA that we want to work with, right? And say maybe there's a EcoR1 site slightly upstream of it, right? So say that we have this double strand here, and this is the region that we want to work with. Well, if there's an EcoR1 site over here, and maybe a BAM H1 site over here, well, we can cut with EcoR1, we can cut with BAM, and now we have excised that region of DNA that we're trying to work with, right? So we can manipulate the DNA in this way. We can cut it out, and now we can do something useful with it. What we can do with it, we'll see in just a minute here. Now, restriction enzymes can be used for subcloning, like we're talking about, right? So we can excise this out and do something useful with this fragment of DNA. We can also use it for analysis, right? Specifically restriction fragment length mapping, right? Or for probing, as you'll see, right? So here, for example, what we have is the genome of a virus, or 2 viruses rather, X and Y, digested with 4 different enzymes, right? So the first 2 lanes are the genomes of viruses X and Y digested with restriction enzyme 1, the X and Y digested with restriction enzyme 2, and so on and so on. And what we can do is we can compare the sizes of these restriction fragments, right? So after digestion, we can run all of the fragments on an agarose gel and separate them by size. Here, the smaller fragments run through the gel faster than the larger fragments, so we have larger fragments on top. And we can see if we compare the genomes after digestion between viruses X and Y, those 2 strains, we can see that they match up exactly, okay? So they're exactly the same. But now if we repeat that with enzyme 2, okay, well, what happens? Well, notice most of these bands are the same, so we have this doublet here, we've got these 2 bands, but interestingly, we're missing this band that is present in strain Y. It's no longer there in strain X. And this band is slightly larger than this band, right, in strain Y. So what does this imply? That implies that in the region, right, where we would find the restriction site for enzyme 2, that has been mutated in strain X, so it's no longer there. So enzyme 2 can't cut at the same place. Oh, I'm sorry. Cannot cut at the same place as it did in strain Y. And that piece of DNA has to go somewhere. Right? And where it goes in this case is still attached to this fragment here. So this fragment appears bigger. This fragment is the size of this fragment plus this fragment. So we have a larger fragment there. And we can see the same kind of pattern continue through enzymes 3 and 4. Notice here we have differences in size. Actually, enzyme 3 is all the same. Okay? And in this way, we can start to put together what we call a restriction map of the genome. In fact, before sequencing was readily available, actual DNA sequencing was readily available, this is how the genomes of several organisms were mapped. Right? We didn't know the sequence, but we knew, using this type of restriction fragment length mapping, right? We knew that gene X was between enzymes 1 and enzymes 2 on chromosome 3. Right? We were able to put together a restriction map that way, right? So we had relative distances and relative positions of all of the genes in the genome. Now, with the advent of DNA sequencing, and specifically next generation sequencing, now this is somewhat trivial, right? We don't do restriction fragment length mapping anymore because we can get the entire length of the genome, right? the entire sequence, so we know the absolute position of the genes. We can also adapt this, right, to probe for whether or not a piece of DNA is present in our sample. So again, going back to our example here, if we know what mutation this is in strain X compared to strain Y, well, now we can probe for that using a method called southern blotting. Right? In this case, we do the same thing. We take our genome, okay, we digest it with a certain enzyme, let's say again, enzyme 2, restriction enzyme 2, because we know that that leads to a differential restriction fragment, right? But instead of just visualizing it using some stain like ethidium bromide or cybersate, which is what's shown here, right? We need a way to visualize the DNA, that's what stain does. Instead of staining this gel, instead what we do is we transfer all of those bands directly onto some polymer sheet, right? Similar to what we discussed for a Western blot, in this case a nitrocellulose sheet. So again, just like a Western blot, we transfer all of those bands onto a nitrocellulose. Now, we add sodium hydroxide, right? What is going to be the effect of adding sodium hydroxide to this sheet here? Now, what is sodium hydroxide going to do in general?
Deprotonate.
Right, sure, exactly. It's going to increase the pH, it'll deprotonate. And what did we learn about the bases, the nitrogenous bases in DNA in chapter 1? No, not negatively charged. The backbone is negatively charged. But they form H bonds, sure. Okay, and what happens to the H bonds as a function of pH?
They break apart.
Why?
Because you're taking away the hydrogen it's using. So
Right. The nitrogenous bases have ionizable protons. So as we increase the pH, we lose those protons, and we disrupt the hydrogen bonding between the base pairs. Right. So we turn double-stranded DNA into single-stranded DNA, which is important for the next step because we're going to add a probe, and we synthesize this probe chemically. Okay, it's just a short oligonucleotide, so it's a short stretch of nucleotides, and we control the sequence because we made it in the lab. So if we're trying to probe for this mutation, right, and we know the sequence of this mutation, well, now all we have to do is add our radiolabeled probe, right, so all the phosphorus in the background have been replaced with P32, which is slightly radioactive. Okay, and what's going to happen? Now it will base pair, if that mutation exists, it will base pair with that mutated DNA sequence, right? And that's what we see up here, right? So base pairs, we wash off all the non-bound radiolabeled probes, so the only thing left should be what? It should be our radiolabeled probe bound to the sequence that it's complementary to, right? So we can expose that to an autoradiographic film and get a band. So if the mutation is there, we'll get a band. If the mutation is not there and we didn't get any base pairing between our probe and the now denatured DNA on the polymer sheet, we won't get a band, right? So we can probe for specific DNA sequences using this method here called southern blotting. Well, now that we've talked about Western blotting also, it's actually a funny story. This was the first blotting technique that was developed. The guy's last name was Southern, so it was called southern blotting. We used to blot for DNA. RNA, right? That same method was adapted for RNA, right? Because RNA and DNA are basically the same, just missing a hydroxyl. So because it's kind of the opposite of DNA, instead of the southern blot, they called it the northern blot. This is the opposite. The western blot came after that, because it's neither DNA or RNA, it's the western blot, right? So all these northern, southern, and western blots kind of play on words. Okay, so now, once we've isolated our,
Question 5 on your slide. If you're labeling the probe to find some mutation, like does that, like how does that work if there are different types of, like you know how we learn about different types of mutations, like point mutations, and friendship mutation? Like how is it an efficient method?
well, all of those point mutations, friendship mutations, all these different mutations, they're still due to a change in DNA sequence, right, between the wild type and the mutant, right? So if you design a probe that can bind only to the mutant or only to the wild type, right? Well, now you'll get binding only when the mutant is present, right? wild type is present, then you shouldn't get binding. And that has to do with how you design the probe. There are some special considerations that you have to take into account when you're designing these probes, specifically to avoid that, right? How do you get it to bind only to a point mutation, which is different only in one nucleotide? Usually you do that by by making sure that the most 3' nucleotide in your probe will match, right? If it doesn't match, then you get much weaker binding. Right? So there's a lot of considerations that go into that. But you can design a probe that can distinguish even between point mutation and the wild type.
Is it more effective, like more time effective to have it just bind to a wild type and then the other probe doesn't bind to wild type? Like you know it's a mutation? Because like, how do you know, like if you're researching for a mutation, how do you know if it's going to get a point mutation for emerging?
Sure, so that depends on the context of what you're trying to do. If you don't know what the mutation is, but you suspect that there is one, then yeah, absolutely, you could design a probe that binds to the wild type, right? And shouldn't bind to the mutant, right? Or you can design one that binds to the mutant and not to the wild type, right? This depends again.
All just changes based on the
Yeah, it depends on how much you know, right, and what you're trying to figure out. All right, so the polymerase chain reaction is actually one of the most important tools that a molecular biologist has. It's actually developed by a grad student. This wasn't his project, but he did write his dissertation on it, and the process is so simple. His dissertation is like 2 or 3 pages. So normally they're 150, 200 pages. And anyways, he's a wild inventor. Anyway, how does PCR work is remarkably simple, right? So we start with our piece of DNA that contains our target sequence. Again, let's assume that it's here, right? The target sequence is in the middle of some DNA double strand. And what we want right now is just more of it, right? There are other applications of PCR, but right now all we want is to take that small amount of DNA, we can start as little as 1 molecule of DNA or 1 sequence of DNA, okay? And we want to make more of it. Well, the first thing we have to do is to heat these to separate them, right? So remember, heat is going to denature double-stranded DNA. So this double-stranded DNA now becomes 2 single-stranded DNA molecules. Okay? To this, we add what we call primers. Okay? And primers are similar to probes, though not radioactively labeled with P32. These are just regular phosphorus. We add primers, but just like the probes, they're short sequences of DNA. They're oligonucleotides that we have synthesized chemically. So again, we control the sequence of these primers in the 5' and 3' direction. These primers we add to this denatured DNA, right? We cool the whole solution down so that the primers can base pair. And we've designed these primers so that they will anneal or base pair upstream and downstream of our target DNA sequence, but on opposite strands. So the forward primer will anneal to this strand, right, upstream of the sequence, and the reverse primer will anneal to the opposite strand, downstream of the sequence. So we have this in the 5' to 3' direction, and this is in the 5' to 3' direction, the opposite way. Now we synthesize new DNA, right, using DNA polymerase. We synthesize this, and it's going to elongate this primer in the 5' to 3' direction because we have a 3' hydroxyl here. And the same over here, it's going to elongate this way because we have a 3' hydroxyl there. So what have we got now? Now we've got our sequence here and our sequence here, right? So we've gone from 1 sequence to 2 sequences. Again, remember, the sequence of the DNA that's going to be synthesized is dependent on the template strand. So this is going to be complementary to this, and this was complementary to this. So this and this should have the same sequence, and the same for the blue primer. So now we have 2 identical sequences. So we've gone from 1 to 2. Now we can repeat this process. Right, so here is where we left off down here, and we begin a second cycle of PCR, but using this as the starting point. Right, so again we heat to about 95 degrees Celsius. We separate out all the strands. We add primers. Okay, so here we have the blue strand, which is the one that we just synthesized. This is going to anneal to the reverse primer. This is going to anneal to the forward primer, and the same down here. And we'll elongate, right? And so what we end up with is this, right? Now we have 1, 2, 3, 4 double strands, all containing identical sequences. We can do a third round now. A third round is going to yield how many DNA sequences? If we have 4 over here. each double strand led to 2 new double strands. So how many sequences are we gonna have? How many double-stranded sequences are we gonna have after the 3rd round?
4.
No, we already have 4.
8.
8. And then 16, and then 32, and then 64, 128, right? So it's 2 to the n. Because in every round, okay, we multiply the number of sequences that we have by 2.
So would it be 2 double strands by the end of the 1st generation? And then that would mean 4 single strands?
4 single strands, right? But this is 1 double strand, and this is another double strand. And we started with 1 double strand. Right? So here we have 1 double strand, here we have 1 double strand, 2 double strands. Here we have, again, those 2 double strands, and we end with 1, 2, 3, 4 double strands. Right? And here, each of these strands, once we denature them, is going to serve as a template. So we'll have 8 double strands. Right? That consists of 16 single strands. So once we denature those, then we'll have 16 double strands, and so on and so on. Right? So here's a kind of a more expanded view, right? So again, we want to denature these. We do that by heating this up to about 95 degrees Celsius. Right? And then we allow the primers to combine. So we have a forward primer binding here, a reverse primer binding here. Right? What's going to happen? This is elongated in this direction. This is going to be elongated in that direction. Remember, DNA synthesis always occurs in the flat prime direction. So now we have these 2 double strands, right? One that was the original parental strand, plus the new strand that came from elongating the reverse primer. And now we have a second, which was the parental strand, plus the strand that came from elongating the forward primer. So now we have gone from 1 double strand to 2 double strands. Now each of these can serve in the next round, right? So we go 4 and then 8 and then 16, so it's exponential. Okay, so again, just more of that. So that means if we started with n DNA molecules, after C number of cycles, we'll have n times 2 to the C DNA sequences, or DNA double strands, right? So a common